and sometimes in in the case of non-coded RNA, which is tRNA, the functional product is functional RNA. SAGE is serial analysis of gene expression. In SAGE is the technique which allows us to rapidly and digital analysis the overall gene expression of a sample or interested cell. SAGE also provides us a quantitative expression profiling to give a given cell population. SAGE technique was first invented and discovered by Victor Wilculus in 1995. At that time, RNA plotting were the technique where to study the gene expression. The technique were slow, so the scientists came up with the new advanced technique, which is more rapid and than RNA plotting, and is able to study the gene expression in a small amount of a time. It allows us to compare gene expression of a normal developmental and disease cell. SAGE also allow us to digitally analysis the gene expression not only of a single gene but a complete profile of a cell gene expression. SAGE is carried out in total 10 steps. First step, isolation of mRNA. The form, from the sample or interested cell or a disease cell, mRNA is isolated. Then from isolated mRNA, we have to synthesize the cDNA. So in that process, reverse transcription is used as an enzyme called reverse transcriptase. Second step, to synthesize the cDNA. cDNA that is isolated from mRNA molecule by enzyme reverse transcriptase. And in that, biotinylate oligo DT primer is added. Biotinylation will help us to isolate the cDNA further in the process. So, cDNA synthesis is immobilized to streptavidin beads. Streptavidin beads is used in the purification of biotinylated molecules. So, cDNA mixed with the substrate of streptavidin beads. These beads will form a biotin cDNA complex, a very strong bond. Third step, enzymatic cleavage of a cDNA. cDNA is cleaved with an enzyme known as restriction endonuclease, which is type 2 restriction endonuclease enzyme. For example, Neisseria lactamica 3, it is a type 2 restriction enzyme. So these will cleave the cDNA into short fragments, and these fragments are of 250 base pairs with sticky ends on the both side. Fourth, ligation of a linker to bound cDNA. The cDNA is ligated with a linker at their ends. The linker is synthetic double-stranded oligonucleotide containing a sequence for a specific restriction enzyme. The linker is added and divided into two groups, Group A and Group B. The linker must contain little nucleotide cohesive overhang type 2 recognize sequence and PCR primer sequence. Fifth step, uh, cleaving with the tagging enzyme. So tagging enzyme is used to cleave a cDNA. So usually used as a BSMFL. It is a tagging enzyme used to cleave a sequence. These remove the cDNA from the build to create a short tag fragment of 11 nucleotide long. These tags have sticky ends which are repaired using DNA polymerase clean off fragments. and DNTPs to create the blunt end tags. Sixth step, formation of a DITAG. After there is a formation of a blunt end tag, the two groups of cDNA are ligated using T4 DNA ligase, and DITAGs are created. The DITAG, a linker DITAG or a linker construct is created. Seventh step, PCR amplification. The linker DITAG or a linker construct is produced. The linker DITAG amplified using PCR primer. Due to amplification, there are many copies produced of the linker DITAGs and or a linker construct of a cDNA. To isolate the DITAGs, the cDNA is again digested by using anchoring enzyme. Anchoring enzyme means it is a restriction endonuclease that recognizes and cut a specific 4 DNA sequence. So anchoring enzyme is then used to cleave the DITAGs and remove the linker. It leads to sticky end on a cDNA. These sticky ends contain a sequence in GTAC at each end of the DITAGs. Contamination of DITAGs. The DITAGs are combined into a much longer molecules on contamers. Contamers means multiple copies of a DNA sequence arranged end to end sequentially. A contamer each DITAG is having a anchoring enzyme site. Anchoring enzyme site allow to compute or uh, monitor and recognize aware of one ends and another begins. A 10th step, uh, cloning and sequencing. Lot of copies are required that the contamers are interested into the bacteria which act like living copies machine to create a millions and billion copies of a DNA. These copies, when they are produced, they are sequenced and stored in Sage database bank. Application of Sage by comparing different types of cell generates profile that will help us to understand healthy cell and what goes wrong in disease. Second, determining a change on gene expression and consequences of experimental treatment. For example, carcinogen and hormones. Third, it involves the identification of differential express gene between two or more states, means a disease or a normal tissue. The goal of a genetic disorder is to illustrate a novel genetic pathway or a causes of specific therapies. Fourth, transcriptomes profiling has been one of the most utilized approach to investigate human disease. Fifth, allow rapid details analysis of thousands and transcript in a cell. Now SNP. SNP is a single nucleotide polymorphism. Polymorphism is a generic term that means many shape. SNP also called in short SNP or point mutation or single base substitution occurring on the genome compared to individual that most of their genome is exactly same and they differ at a certain place. So these certain place are covariance. Covariance means how two variables are related to another, and one of the form of these variants are single base substitution or single nucleotide polymorphism or a SNP. It is a DNA sequence variation occurring when a single nucleotide A, T, C or G in a genome differs between a member of a species. For example, in the picture, two sequences sequence DNA fragments from different individual AA, CG, TA to AA, GCTTA. Container differ in C and T. It is a single nucleotide polymorphism. These are not just one SNP whenever it is compared any two individual, but there are millions of these SNPs, and these SNPs are used as a genomic marker throughout the genome. Genomic marker its position where it is located exactly on genome, and genotypes can be associated with further traits of interested or use them while searching for a specific gene. Millions of SNPs in any two genomes are interested in genotyping of these chips, called as SNP chips, also called as a microarray or a bead array. For genotyping, SNP on the SNP chip are aligned by a design, means they have exactly two alleles that translate three genotypes. Also step can be missing. For example, there are A and B. Then genotype would be either AA, so it is a homozygous, AB heterozygous, or BB other homozygous. Genotyping data look like this. So genotype file at the end, huge text files, but the important thing, a genotype that is located in a particular data. Application of SNP. First, uh, it involves the identification of a differential expressed gene between two or more states. Second, they involves analysis polymorphism variants in candidate gene with a known role in biology, and relating carriage of a specific allele to quantitative trait or a disease of a interest. Fourth, response prediction. Fifth, to identify the mutant genotype responsible for the inheritatory genetic disorder. Thank you.
Okay, thank you very much. Very good presentation. उपस्थित असल्यापैकी कुणाला काही प्रश्न विचारायचे? कविता, दीप्ती, अनिकेत.
नो सर.
सर फक्त आता ते
अनिकेत आवाज येतोय मला?
सर ते फक्त तुम्ही आता शिकवणार नाही ना, हे डबल झालेलं सेमिनार घेतलेले टॉपिक?
नाही 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 नाही. का रे, काय शंका होती का कशात?
नाही सर, मग ते जे जे टॉपिक घेतात ना त्यांना फक्त
हॅलो?
पीपीटी शेअर. हॅलो.
हा, सांगितलंय ना मी.
हा
पीपीटी
तर
टाकणार
म्हणजे
आहे. हा,
हा
सांगितलंय. येस, ह्याचं लिटरेचर आधी पण मी थोडंफार पाठवलंय, पण आपापल्या कविता.
हा सर.
दीप्ती, पीपीटी
हा
पाठवा ना
सर.
तुमच्या बाळांनो.
हा पाठवतो. हा सर,
हा
आज पाठवतो. हा पाठवतो.
हा, किती पण पाठवा. ओके, अनिकेत तुमचं झालंय सेमिनार?
सर नाही झाला. नेक्स्ट टेस्टला दिलं तर चालेल सर.
बरं, कधी
सर
देताय?
पीपीटी राहिली सर माझी करायची.
बरं बरं, ठीक आहे ठीक आहे ठीक आहे, नो प्रॉब्लेम नो प्रॉब्लेम. ठीक आहे, मग अजून काय शंका आहे का आतापर्यंत झालेल्यामध्ये कोणाला काय? हॅलो.